Hello and welcome to the eOrganic webinar on assessing nitrogen contribution and Rhizobia diversity associated with winter legume cover crops in organic systems, presented by Julie Grossman of North Carolina State University. My name is Alice Formiga and I'm the webinar coordinator for eOrganic. eOrganic is the organic agriculture community of practice with eExtension. We're a community of cooperative extension service personnel, researchers, ag professionals, organic certifiers, and practitioners. This is the third webinar in this winter's cover crop webinar series. You can find all of our recorded webinars, information about upcoming webinars, as well as articles and videos on organic farming on our website at extension.org/organic_production. We're very glad to have Julie Grossman with us online today. Julie is an assistant professor at the Department of Soil Science at North Carolina State University. Her work broadly explores the ways in which we can better manage plant-soil microbe relationships to enhance soil fertility, with the ultimate goal of developing sustainable food production systems. The overarching goal of her research program is to enhance the efficient management of soil nutrients in low-input and organic farming systems. She also served on the steering council of the Sustainable Agriculture Education Association, a new professional association championing innovative educational approaches for sustainable agriculture. After Julie's 45-minute presentation, you will have the chance to ask her questions. If you have a question, you can simply type it into the question box on your screen and hit return. If you can't see the question box, click the small plus sign next to the word question on your control panel to open it up. We'll answer as many questions as we have time for in the 30-minute question period following the presentation. Now I'm going to switch control of the screen over to you, Julie.
Great. Thank you, Alice. Well, it's really exciting to have so many people out there who are as excited about cover crops as I am. We're going to start today by, by me getting used to my screen. There we go. Start today by talking a little bit about what nitrogen fixation is and how it works. And then we're going to spend a majority of the time talking about some of the research we're doing here at North Carolina State on legume cover crops. And if you're on this call, you're already very familiar with some of the benefits of using legume cover crops, including weed suppression, pest control, and increase in organic matter and soil nutrients. And in particular, we're going to talk about how cover crops can contribute new nitrogen, so fixed nitrogen, to your farming systems through the process of nitrogen fixation. Okay, so here on the right you see me, and behind every PI is, of course, a group of wonderful, amazing graduate students, and this is my herd to my right, your left. And the research I'm going to discuss today has primarily been done by these two women sitting next to me. This is Mary Parr, and this is Napi Mothapo. Okay, so cover crops, they're not just a good idea, of course, but the National Organic Program standard actually requires that they be used in organic production, stating that organic producers should manage crop nutrients and soil fertility through rotations, cover crops, and the application of plant and animal materials. And in the current climate, it's really likely that in the coming years we'll see increases in the cost of fertilizer nitrogen, conventional nitrogen, which make legume cover crops a good bet for not only organic producers but conventional producers. And cover crop economics are rooted in the idea that as a legume gets bigger and acquires more fixed nitrogen, so more overall nitrogen, that you can reduce the costs of nitrogen inputs that might need to be purchased from off-farm by using that fixed nitrogen from the legume. So we're going to talk a little bit about that today. So today I'm going to really discuss the process through which legume cover crops bring new nitrogen into your farming system and provide you with some recent data from our lab regarding both nitrogen fixed by a variety of legume cover crops and the Rhizobia that are associated with these cover crops. And I do have a goal for you. I want you to be able to leave this talk today with some tools you can use on your own farm in order to manage your legume cover crops. So these tools might be foundational knowledge that you can use to design your own mini experiments, or they might actually be things you can measure and learn more about nitrogen fixation by looking at and measuring your cover crops. So legume cover crops can be used at a number of different points in the growing season. Primarily here in North Carolina, they're grown either during the summer fallow or the winter fallow. And today I'm going to be talking about cover crops that we're using, legume cover crops that we're using during the winter fallow. So they're planted in the fall, they go dormant, grow a little bit, go dormant in the winter, and then they're terminated in the spring before the cash crop is planted, either through incorporation, mowing, or herbicide. And some of the research I'm going to talk about today is I'm looking at a fairly new way of terminating the cover crop, which you've heard about in previous webinars, the roller crimper. So that nitrogen, after the crop is terminated, is then mineralized or released for the cash crop to then use. And in legumes, they carry out the process of biological nitrogen fixation. So what is nitrogen fixation? Well, this process it's an association between plant and soil bacteria, where they're both getting something beneficial from this relationship. The soil-dwelling bacteria are called Rhizobia, and they live in ball-like root structures on the legume and fix or convert atmospheric nitrogen, which is otherwise in a form that's unavailable for plants to use, into a form that plants can use. Okay, now I'm gonna ask you guys a question. You should have a poll that appears on your screen. I want to know how much of the atmosphere is made up of nitrogen. I'd like you to select one of the following. It's either 10%, 50%, or 80%. How much is made up of nitrogen? Okay. Hopefully you've all gotten a chance to attend. It looks like I have to take it too. Let's see. Oh, okay. Okay, so the answer to that is, of course, 80%. It's almost 80%. So a good percentage of the nitrogen that's in of what's in the atmosphere is actually nitrogen, but it's completely unavailable for plants to use. It's in the wrong form. It's very, very tightly bonded, and the only way to break those bonds and to make that nitrogen available is through either the Haber-Bosch process, which is high pressure and heat used to make nitrogen fertilizer, or through this process, this biological process of nitrogen fixation. So there's a little bit of a misconception I found when thinking about nitrogen fixation, which is that the nitrogen actually is leaking out through the root nodules into the soil with the fixed nitrogen. Then it becomes available for other plants to use. And that's not correct. So I want to spend a minute here just describing this process and making clear that when these nodules that are in contact with soil atmosphere, so in the pore space, the rhizobia and the nodules fix the nitrogen, which then the plant uses. So it goes up into the shoots of the plant, and that plant will use it for its own growth and development. In return, the plant will contribute carbon to the rhizobia living in the roots. So those and the bacteria in the roots, the rhizobia, use the carbon for their own growth and development. So that's why this is a mutualism or a symbiosis, the beneficial relationship for both partners. When this crop dies, legume crop dies, and is decomposing in the soil, that fixed nitrogen is then released into the soil and can be taken up by non-legume plants such as corn. So that's the process. Without that decomposition of the legume, nitrogen would not be made available. It just stays trapped in the organic material of the legume. So this picture here I thought was pretty interesting. It's showing rhizobia as they're about to associate with a legume. So they're gathering around the root and getting ready to form the nodules, form the balls through which the plant will curl around the bacteria and create basically a new structure in which the bacteria are housed. Nitrogen fixation occurs in a number of different bacteria, but here in this seminar today we're only going to be discussing the group that are collectively known as Rhizobia. So you may in your studies throughout the future hear about other types of nitrogen fixation which do exist. It happens with free living organisms as well as other plants. But here we're only going to be talking about the association of Rhizobia with legume family plants. Here we have what a root system of hairy vetch looks like, the well-nodulated hairy vetch. You can do this yourself on your farm. You can do it by pushing a shovel into the ground about 6 inches down around the perimeter of the root and gently prying that root system up. The reason you don't want to reach down and just grab the shoot and pull the legume out by the shoot is that you'll scrape off the nodules. They'll be stripped off through that pulling action. So you pry them out gently with a shovel. You can rinse them in a bucket and take a peek at how many nodules you have on your legume. A well-nodulated legume is not alone indicative of good nitrogen fixation, but what you can do then is take a razor and cut open one of those nodules. If it's pink inside, that means there's an actively fixing bacteria present in the nodule that's fixing nitrogen for that crop plant. There are a number of factors that determine how much of this free fixed nitrogen is given to the legume through nitrogen fixation. Number one, there needs to be an effective symbiosis between the soil bacteria and the legume. If you don't have the right type of bacteria present, or there's no bacteria present in the soil at all, then you won't have nitrogen fixation. Second, there need to be environmental conditions that are conducive to the survival of that Rhizobia. So pHs lower than 5, for example, are not conducive to Rhizobia survival. Your Rhizobia are not going to live in that environment, as well as having waterlogged soil. Rhizobia need oxygen to survive, and you need to have a fairly well aerated soil in order to promote that survival. And finally, the native nitrogen fertility of your soil is also a factor that will determine how much nitrogen fixation is going to take place. So right now I'm going to show you a couple slides that are borrowed from the Drinkwater Lab, Lori Drinkwater at Cornell University, which is where I worked for a couple of years, looking at how the native amount of soil or nitrogen in a soil will impact the amount of fixation that occurs. It's well known that if given a choice, a legume is going to take nitrogen from the soil as opposed to fixing it themselves. Now, why would this be? This occurs because it's very, very expensive for a legume to fix nitrogen. They can spend about 20% of their carbohydrates. If you think of their carbohydrates as being money, they can spend about 20% of the entire carbohydrates that they have made through photosynthesis on hosting that bacteria, that Rhizobia bacteria. So they give the carbohydrates to the bacteria, and the bacteria gives back nitrogen. So we talk about it as being free to us, money-wise, but it's not necessarily free to the plant. So if this legume is planted into an environment, if this is the soil nitrogen pool, where there's a little amount of soil nitrogen available, it's going to fix more nitrogen. See, the size of that arrow is much bigger than the size of that arrow. Where if you have a legume that's planted into an environment where there's lots of available soil nitrogen, it's going to take up more nitrogen from the soil because it's cheap, rather than fix it themselves. And so it's been hypothesized that over time, especially in organic systems where you're creating this reservoir, this pool of organic nitrogen that's going to be made available over a period of years, such as compost, that fixation will decrease. And this hypothesis was tested by Megan Shapansky, and this was just published in Plant and Soil in 2010, where she looked at farms that had been under organic management for up to 20 years. And she looked, first measured the amount of total soil nitrogen in their soils, and found that, yes, there was an increase. That as farms had been organic for longer, or under organic management for longer, they had increased their soil nitrogen. She then looked at soybeans planted into those soils and how much nitrogen they were fixing in each scenario, and found that as years under organic management increased, the amount of nitrogen being fixed decreases. So that demonstrates how the native amount of nitrogen you have in that soil can impact the amount of nitrogen fixation that's occurring. And just to put it in context, I think a lot of you probably have seen this before. It's just a typical rendition of the nitrogen cycle. And I wanted to quickly go through it and show you how nitrogen fits into this, how nitrogen fixation fits into this overall cycle, where we have nitrogen being fixed from the atmosphere into the legume then decomposing in the soil and becoming organic matter, organic material, and releasing available forms of nitrogen into the environment, such as ammonium and nitrate. And then those forms of nitrogen are available to be taken up by plants. So the timing of the release of nitrogen from legume cover crops has been highly studied over the last 50 years or more, where we're really interested in trying to create a synchrony between the rate at which legumes are releasing nitrogen into the soil and the rate at which the crops are taking up or have a need for that nitrogen. So here you see an example of what it would look like to have an asynchrony or a lack of synchrony, where your cover crop is incorporated, it's fixed a lot of nitrogen, and it starts to release that nitrogen through the decomposition process, having the height of release right here, but the crop demand doesn't happen until much later down the road. So this has been an ongoing challenge for those who are managing soil fertility for organic management to try to match that crop demand with the rate at which legume cover crops are releasing the nitrogen or mineralizing that nitrogen into the soil. So in our lab, we try to take what we call a systems approach to managing nitrogen in organic systems, and we focus almost entirely on organic systems, where we look at the legume and the amount of nitrogen coming into that legume through nitrogen fixation. We then look at the rate of decomposition and different ways to terminate the cover crop and how that would impact the amount of soil nitrogen that's released and the rate at which it's released. And we look at the uptake of that nitrogen into primarily grain systems to try to get a sense of that synchrony, if we're having the nitrogen be released at the same time that the crop needs it most. And a big area of emphasis that we focus on in our group is looking at the Rhizobia ecology and trying to create a symbiosis that works well. So trying to get the right Rhizobia in association with the legume cover crops so that large amounts of nitrogen are being fixed with that overall goal of having a lot of biomass, a lot of cover crop growth that has a large amount of nitrogen present in the biomass so that when it decomposes, it's present for other crops to then take up. So we're going to focus today on nitrogen fixation of some legumes that we've been looking at here in North Carolina, the decomposition of those cover crops, not all of them, but just a couple of them we're going to look at today. And finally, I'm going to end with talking a little bit about what we've learned about Rhizobia ecology, specifically looking at Rhizobia that are associated with hairy vetch here in the state. So I mentioned earlier that we were focusing on a no-till system of termination, and I know a lot of you have heard about this, the roller crimper, which kills the cover crop or rye or other usually grass or legume cover crops by rolling over it with a large water-filled drum. And this has to happen when the cover crop is at full maturity in order to kill it. So we have used this over the last couple years in managing legume cover crops, where we plant the cover crop in the fall, let it grow a little bit, it puts out a lot of growth in the spring, and then in April or May we have rolled over it with at 3 points in time to try to determine what the timing is for being able to kill these cover crops, and then tracked the release of nitrogen from these cover crops into the soil. Because if you think about it, if you look at that mulch that's being created into which we're going to plant corn directly, basically on the same day it's rolled, you can see that it creates a very thick layer of dead vegetative material. And in the decomposition of this material, there's only going to be a small layer of it that is in contact with the soil at any one time. So decomposition, theoretically, we thought might be a little slower in these systems and try to get it to match the uptake of the corn. So the main questions we're focusing on in our group include we want to know how much nitrogen is in the legume biomass and how much of this is contributed through biological nitrogen fixation. So if you have a total amount of nitrogen, some may have in the crop, in the legume, some may have come from the soil, but another percentage of it likely is from nitrogen fixation. So we're trying to tease out how much is free nitrogen from fixation versus nitrogen that's been taken up from the soil. Secondly, we're interested in how nitrogen is released from this decomposing mulch, and in particular, the research I'm going to talk about today, we're looking at mulch that is left after it has been rolled with the crimper. And finally, we're interested in the diversity of Rhizobia that are able to nodulate and fix nitrogen with hairy vetch here in North Carolina It's a very popular cover crop in the area, and we'd like to get a sense of who's actually in the nodule when farmers are planting this on their farms. So we've looked at 14 different legume types. We have a number of different species, and within each some different cultivars. So within this hairy vetch, we have 4 different varieties that we're studying, including 2 varieties out of Auburn, AU Merit and AU Early Cover. We also have a variant of the early cover from Auburn that we call Winter Hardy Early Cover. And this is basically a hairy vetch type that has been selected by Steve Groff from Pennsylvania to be winter tolerant. Since the early cover is primarily for southern environments, he had to go through a number of years of selecting for a variety or type of this early cover which could withstand the winters. And finally, we looked at Purple Prosperity, which is a newly developed cultivar out of the Sustainable Agriculture Lab at USDA ARS, designed to be early flowering in order to capture that height of nitrogen fixation that occurs when that plant is starting to flower and be able to get a crop in the ground earlier in the season. We also looked at 4 varieties of crimson clover. We have 2, again, from Auburn, and 2 more, Dixie and Tibby. And then we looked at a variety of forage legumes, including balansa, berseem, subterranean clover, and lupin. And we looked at 2 varieties, or 2 types of Austrian winter pea, Whistler, and then just the regular variety unstated. So I'm going to talk a little bit about those today. And it's nice to see the list to be able to describe them, but to me, it's really, of course, more beautiful to see the pictures. And our fields, when they're in bloom, are just gorgeous. It's this patchwork of color from all the legumes that we're looking at. And here you can see a few of them in this slide. I mentioned we had 3 termination dates. So the first year we carried out this study, we tried to roll them really early. We tried to roll them in mid-April. And basically it was not successful. We didn't have very good kill of the cover crop, but it ended up competing with our corn quite heavily. So for the first year and the second year, we concentrated on these what we call mid and late termination dates, where we rolled them in early May and late May. And again, when I say roll, we're rolling over each of the different cover crop types that I showed you in the previous slides. This picture is showing my technician, Sarah Seehaver, cutting out a half-meter squared quadrat of biomass from the plots. And we basically would take that biomass back to our lab and do a variety of things with it to measure how well that cover crop grew. And again, this is the research that's being done by Mary Parr. You can see her picture in the upper right-hand corner there with a nice corn necklace on. You get to do all kinds of fun things when you're working out in the field in the summer heat of North Carolina. So she was really interested in how much nitrogen overall was taken up in the cover crop biomass, as well as how much nitrogen had been fixed. So she looked at percent total nitrogen in that biomass, the amount, the weight of the biomass nitrogen. And she also looked at using isotope ratios, the amount of nitrogen that came from the atmosphere, which is, of course, the amount that was fixed in that cover crop residue. So how much biomass is actually contributed from legume cover crops? Well, you can see here all the cover crops listed on the x-axis and the mass of those on the y-axis. And you can, the megagrams is just it's 1,000 kilograms per hectare, which is a little more than pounds per acre. So you can just, if you're thinking in terms of pounds per acre, you can just reduce it by a little bit in your head. Here we see hairy vetch was one of our big winners. It produced about 5,000 megagrams per hectare. I'm sorry, 5,000 kilograms per hectare of biomass to the following that roll. And the rye mixes, if we look to the end, these are something I didn't mention in the previous slide, but this was something we did in just the most recent year to see how some of the cover crop mixes, if we mixed it with a grass and a legume, how they would perform in a rolled environment. So MXE, you see here, this is this is the early cover vetch with rye. This is Merit cover hairy vetch with rye, and then we have an Austrian winter pea mixed with rye. And so we saw that on average the the merit vetch, when grown with the rye, really had a lot of success, and I'm going to show pictures of what that looked like later on in the presentation. Here we can see the nitrogen content of the legumes, and this is, of course, what we really want to know from my end of things. How much nitrogen was being contributed to the system following rolling of these cover crops. And again, this was total nitrogen contained in the cover crops. So it's not looking at how much nitrogen is actually fixed at this point. It's total nitrogen. Nor is it looking at how much nitrogen is released into the environment following roll down. It's that cover crop needs to be mineralized through microbial action, released slowly over the growing season into the environment. So here, again, you can just think of it, if you're thinking in pounds per acre, you can just decrease that by a slight bit. So we have the hairy vetch coming out on top, as always, where they produced a good amount, about 130 pounds of nitrogen per acre. In some of our sites, P a little bit lower. And crimson clover, we found that it had a very early mature date. So by the time we got to our second roll date here at the late roll in May, that plant had probably channeled a lot of its nitrogen into seeds, and when it was rolled, there wasn't a lot left in the tissue, which is what we were primarily quantifying. So we had a great reduction in nitrogen, but in this species at the late roll date. But on average, we felt we had pretty good nitrogen being contributed from these cover crops, and in some cases it was enough to take care of, at least in our corn, to take care of a lot of the nutrient requirements of the corn in that growing season. So I told you in the beginning that I was going to give you some tools you could use on your farm to do this sort of thing. And so the answer to the question of can I do it on my farm? Yes, you can. You can figure out what percentage of nitrogen is in your cover crop biomass, and then by scaling up to the size of your field, you can determine how much nitrogen you're actually putting onto that field in the legume. So there's some basic steps you can do on your own to make this happen. One would be finding a frame, such as what we do, or a yardstick that's of known dimensions, and then clip several areas of your field. So you clip, just like you were seeing Sarah do in the earlier slide, you can clip it down to the ground, and then estimate dry the legume biomass. You can put it on the dashboard of your truck, you can put it out in the sun, you can dry it in a low temperature oven until it's crunchy dry, so very dry, and then weigh it. And from that point you can determine the amount of biomass you're adding per acre by using the following equation. You use the total weight of your dried samples, and you divide into that the area that you've sampled, and we're going to work through a quick example in a minute. And then you take this value times the amount of square feet that are in an acre, and it'll give you the amount, the pounds of material you're contributing per acre. So let's work through that together. Okay. So let's pretend you've sampled two 2-by-2 foot regions of your field, and the dried samples together weigh about 0.5 pounds. So you want to determine how much biomass per acre you have. Okay. So if you look down below here, you see how we worked this out. The answer is you're contributing about 2,700 pounds per acre. So you've got 4 square foot areas. So basically you have 8 square feet that have been sampled. And you know that together, all the biomass you've collected comes to about 0.5 pounds. You take that times that 43,560 square feet, that's in 1 acre, and you come up with this final value of having 2,722 pounds of material. This doesn't have anything to do with the nitrogen yet, pounds of material on your farm. So how do you calculate the nitrogen? Skip that for now. The nitrogen is calculated by either using a best guess estimate, or by sending off a tissue sample to your local extension service. I know here in North Carolina, you can pay about $5 and take in some tissue from your field and get it analyzed for nitrogen content. And I assume that that's probably a possibility for many of you elsewhere in the country as well. But in general, cover crops by the time, or legumes, by the time they flower, they contain between 3 and 4% nitrogen. So of that biomass that you've collected, 3 to 4% is nitrogen. So let's pretend that we take our 2,700 pounds of material and we know that it has about 4% nitrogen at the time we've killed it. How much nitrogen are we applying to our field? Well, we're applying about 108 pounds of nitrogen. Now, how did I figure that out? Well, I take that 2,700 pounds and I multiply it by 0.04. So that's, you take that times the percentage and you get about 108 pounds per acre. Now, as I mentioned earlier, this is not the total amount that's being added that will be available immediately. It needs to be acted upon by microbes in the soil that will then break it down and make that nitrogen available to your subsequent crop. So Mary, when she did her research, she was really interested in what the timing of optimal nitrogen was in her system. So what you're seeing here are the different categories of cover crops that we looked at. We have pea, vetch, crimson clover, and some of these forage legumes. And in each of the squares, and the quadrants you see here, we have 3 roll dates. The early roll date of mid-April, early May is in the middle here, and late May was the last roll date. And on the y-axis, you have the kilograms of nitrogen per hectare that was produced by that cover crop. And you can see that for us here in North Carolina, it was really that early May date for almost all of these species and varieties that was the real winner in terms of delivering nitrogen, the optimal nitrogen to our systems. The one exception is up here with the vetch, where we look at the early cover vetch, which was at its peak nitrogen much earlier in the season. About mid-April, it had reached peak nitrogen and then began to decline. By the time we rolled it in early May, it had declined quite a bit. And again, this is that variety that was developed for the southern climates and was intended to be something that would develop much faster and put on a lot of its nitrogen earlier in order to get crops into the ground following termination of the vetch. So how much nitrogen was fixed in these species? This was really, I was trained in a nitrogen fixation lab and was really excited about learning a little bit about how much nitrogen is being fixed in some of these species, and some of them had really never been looked at before. And what we discovered was that it depended largely on where they were grown on our field sites. So we had different rates of nitrogen fixation depending upon where we planted, and that makes good sense because we're going to have different backgrounds of soil nitrogen present at these different sites. So if we look at these charts here, you can see the same designations for the cover crops along the x-axis. And then on the y-axis, the nitrogen that was contained in the biomass of the different cover crops, breaking it into the amount that was taken up by the soil and the amount that was fixed. And if you remember, a legume can have both types of nitrogen in it. It can take up nitrogen from the soil as well as from the atmosphere. So at this site, we found that the majority of the nitrogen contained in the cover crop was actually from fixed nitrogen, whereas in this other site in the Piedmont area this year, which was our top performing site out of 6, I should mention we have 6 different sites, we found that while the nitrogen fixation, nitrogen in the plant, about 60 to 90% of it was provided through fixation, with the remainder being taken up from the soil in that light green amount. So when we think about what's going to happen then to that fixed nitrogen, we know that in plants, once you have a plant that's decomposing in the soil, the rate at which it decomposes and releases its nutrients is very much determined by the quality of that plant material. And what I mean by quality is the amount of carbon to nitrogen, so the carbon to nitrogen ratio, which is just a determination of how much nitrogen relative to the total carbon is contained in that crop plant, and other things like polyphenols and lignins. And when we did a determination of the C to N ratio in all of these species that we looked at, and we found that we did have some clustering of the vetches and the crimson clovers together as having very similar C to N ratios, carbon to nitrogen ratios in their tissue. In general, you can think about the rate at which that nitrogen or those nutrients are going to be released as related to these C to N ratios as being if there's low nitrogen in that plant, so mostly carbon, so higher ratios, you're going to have the cover crop decompose and release nitrogen at a much slower rate. And if it's above 20, 24 C to N ratio units, then you have sometimes a net immobilization where the bacteria are trying to get nitrogen out of that material. They can't do it, so they end up taking it from the surrounding soil. So that's net immobilization. It will make that nitrogen unavailable to your subsequent crop plants. But down around the range of the hairy vetch, so we have here between 12 and 15 or so, there's going to be a net release of nitrogen into the environment. And so we sense that there might be a difference in release rates of nitrogen just very much based on the C to N ratios of that material at kill time. And you can also see that the C to N ratios did change from one termination date to the next. So as the crop got older, the late termination date, you can see an increase in both the vetches and the clovers. It's had a greater increase of that C to N ratio. And we did have some differences within the mixes as well, where some of the mixes, in this case the P, had a particularly high C to N ratio at kill time relative to the vetches, the XMM and XME. So this is what it looked like. This was about 2 months after we had killed the cover crop with the roller and planted the corn. You can see it's a very thick mass of dead cover crop material on the surface, and you can see that there are some weeds popping through. The original intention was to have so much biomass be produced that we would have ample weed control as well. And in some cases that did happen, but in others we just didn't have enough biomass to control the weeds. So I mentioned that we were also interested in looking at the nitrogen that's released from the cover crops. So I'm going to talk to you briefly about how we measured that. And we didn't look at all the cover crops. We only were interested in one variety of the hairy vetch, that being the early cover, and the sunrise variety of the crimson clover. So of course the first step is to roll the biomass. In this case you're looking at rye, and what rye looks like when it's rolled, but I'm taking my quadrat sample right there. Step 2 is to use these ion resin probes that basically attract and suck out all of the released nitrogen and hold it on this resin until we can take the probe out of the ground and measure it. So we pop these things in every 2 weeks and then take them back to the lab. We actually send them away to a lab and have them measured to look at between when we put them in the ground and take them out 2 weeks later, how much nitrogen has been released into the environment, which is, of course, a measure of how much nitrogen we hope will then be available for the subsequent corn to take up. We also took traditional soil samples at the same time and compared our results to this point-in-time measurement of how much nitrogen is in the soil at the time of sampling, compared our flux or our unit of nitrogen release over time to the point-in-time measurements of the soil samples. So about maybe halfway through the growing season, we started to notice some differences between the growth of the corn planted into different cover crop mulches. And in particular, we noticed that the corn that was planted in the crimson clover mulch was suffering from some nitrogen deficiency, where it was much yellower than, say, the corn that was in the hairy vetch mulch. And also, which is not shown in this picture, corn that was planted into a control plot that was given about 150 pounds of nitrogen per acre, so a positive control. And so we started to realize that, well, there's probably something going on here in that the rate of nitrogen is being released to the corn growing in the the crimson clover mulch. So what I'm going to show you now are some some data looking at the date on which we were pulling those probes out of the ground or taking our soil samples. Here on the bottom is the probe data, and on the top you can see the traditional soil sample, soil core data. And you can look at the trends over the course of the growing season of how much nitrogen was released, with the green in both graphs being the cover crop residue and the nitrogen released from the cover crop residue, the yellow being the hairy vetch, the red is the zero nitrogen control, so this did not receive any nitrogen, nor did it have a cover crops previously growing on it prior to roll. And then finally, the positive control, you see it depicted here as 168 kilograms of nitrogen per hectare, which is about 150 pounds. And you can see that an interesting thing happened. We had the crimson clover nitrogen being lower than the zero nitrogen control, which was basically just a measure of the nitrogen that was being released from the native organic matter in the soil. So what this is telling us is that there was actually nitrogen being removed from the soil when there was crimson clover mulch present. And this does make sense, because if you think back to when we were looking at the plant quality measurements of C to N ratio, we had very, very high C/N ratios in that crimson clover mulch, which might give us an indication that we would see some net immobilization taking place. The other thing to notice here is that the crimson clover, or the hairy vetch mulch did have a nice release of nitrogen. At some points it was about the same as the positive nitrogen control, and it wasn't until we added that split nitrogen application after the start of the growing season that it shot up in both cases. But we did have pretty good release from the hairy vetch cover crop in these systems. Okay, so now I'm going to show you some pictures of Jonathan. Everybody meet Jonathan. He was our undergraduate student extraordinaire who slaved away in our research plots last summer in the hot North Carolina sun. And we put him here because we wanted to allow viewers of these pictures to understand how tall the corn was. And he's a pretty tall guy. He's like maybe 6 feet tall. But in this picture here, you're looking at what the corn looked like when it was grown in the crimson clover mulch. Then this next picture, this is actually the vetch, the early cover vetch. Doesn't seem that much taller than the crimson, or maybe a little bit darker. Next picture, we have the zero nitrogen control. So you can see a little bit of the color difference between the early cover vetch and the zero nitrogen. Here we have the positive nitrogen control, the 150 pounds of nitrogen application, followed by the mix. I don't know if you remember back to that data I was showing you where we had the rye with the vetch mixture, and we had very, very good biomass coming from this mixture. It was quite impressive. So let's jump for the last little bit of this talk. I'm just going to talk briefly about inoculation and Rhizobia ecology in organic systems. So inoculation is this process of adding to your seed at planting the right Rhizobia. You can look it up in a book, you can get recommendations from your seed supplier to get a sense of what Rhizobia should be present with that particular host that you're planting, whether it be vetch, crimson clover, Australian winter pea, what have you. So a recent survey that was conducted here by Suzanne O'Connell, who's a PhD student in the horticulture department at NC State, found that only about 46% of farmers that were interviewed that were using legume cover crops were using inoculants regularly. So I'm going to do the same thing with all of you out there, and I would like to ask if you inoculate your legume cover crops on a regular basis. And I understand this might be a complicated question, because some of you may inoculate under certain conditions, but just give me a sense of whether you, in general, inoculate your legume cover crops. Okay. So John, if you can take me back to my presentation now. There we go. Oh, there are some results. I don't know if everybody else can see the results, but you can see we have about half of you, that 47% that always inoculate or inoculate on a regular basis, and 53% of you that do not inoculate on a regular basis. So you can see that the research that was done here in North Carolina may be true nationally as well. So Rhizobia that you have in your soil can really come from one of two sources. It can be, or I'm sorry, the Rhizobia that are in the nodules can come from one of two sources. It can come from the inoculant that you've added at planting, or it can be an indigenous or naturalized Rhizobia that's already present in the soil at the time of planting. So these native bacteria, or the ones that are in the soil already, can be beneficial because if they fix large amounts of nitrogen and you don't have to inoculate, it's saving you time, it's saving you a little bit of money, not a lot of money because inoculants are cheap, but inoculation is a process. It does take some time to inoculate these seeds. So they can be beneficial if the Rhizobia that are already in the soil are useful, but it can also be extremely problematic if these native bacteria are competing with an inoculant strain that you've added that might be better and are less efficient at fixing N. So we're very, very interested here in trying to determine who is getting into the nodule at planting. And in particular, we're looking at this type of Rhizobia called Rhizobia leguminosarum, which is the Rhizobia species that is associated with vetches, peas, and lentils. So in this case, it's the Viciae group that is associated with hairy vetch. So we are taking a survey of our own to determine not only how many people are inoculating, but how people are taking care of their inoculant prior to inoculation. We want to know how it's handled before you actually inoculate the seed, how long it's stored, things that might make the inoculant that you're putting on either viable by the time it's put on at planting or not viable. Maybe the majority of the bacteria in that inoculant are dead. So we have a survey that we'd like to put forward to you guys if you're interested in taking it. And there are 3 prizes available. One is this popular SARE publication, Managing Cover Crops Profitably. The second is Crop Rotation on Organic Farms, and we do have a very special version of this where it's been signed by Chuck Moeller, one of the authors. And the best of all, an NC State agriculture hat, and I know that's the one everybody really, really wants, right? But if you volunteer to take our survey, which we think will take about maybe 5 to 8 minutes to complete, we will enter you in this raffle. So if any of you would be willing to take this survey, you could just say yes on this poll, and we will get your email address, and when the survey is released in the beginning of the year, you'll get an email that will bring you to a web link, and then you'll be entered in that survey, or excuse me, in that raffle to win some of those just fabulous prizes. So the Managing Cover Crops Profitably book is a fabulous one. It's really, really good. I use it all the time in my own work, so it's highly recommended. Okay, thanks everybody. Moving on, we just briefly wanted to present to you some of the research that we're doing in looking at Rhizobia diversity across the state, and in general we're finding that when looking at vetch, that there are completely different populations in some cases of Rhizobia, the kind of Rhizobia that nodulates vetch, present around the state. Where here we looked at 3 sites, and one site had almost an entirely different type or suite of Rhizobia bacteria that were present in the soil compared to 2 other sites that were studied. And we're using molecular techniques to analyze these Rhizobia, where we actually are taking them and subjecting them to a number of analyses in our lab where we learn about their genetics and how they might differ from one another. The student that's working on a lot of this work is Napi Mothapo, and she's trying to determine in particular how history of hairy vetch, so how planting hairy vetch in your field might influence the ability of a future legume to nodulate. And so she's looking at paired sites across the state that will determine this, or help us answer this question. There are a number of management practices that can have an impact upon Rhizobia diversity. It's been shown that monoculture of a particular crop over many years in combination with synthetic or chemical inputs can have a great effect on Rhizobia diversity. And we hypothesize here in our group that there are a number of factors that are unique to organic management that will impact the efficiency and effectiveness of Rhizobia in these soils, particularly things like history of a legume being planted in a particular field, the introduction of new Rhizobia into a field, which may have had many other legumes present in the field in the past, and of course a variety of carbon inputs that are used in organic management, such as manures and compost. So NAPI is looking at 3 sites across the state, and each of these, she's gone to organic farms at each of these locations, and she has selected 2 fields, one which has a history of hairy vetch cultivation and one that does not. So each of these sites has a positive and a negative history of vetch cultivation. In each of the sites, or 2 of the sites, they've used vetch in the field continuously for over 20 years, and in the 3rd site, it's over 10 years. And in the fields that are the same soil type on each of these locations, they haven't seen vetch in 15 to 25 years. So what we're doing, or what NAPI's doing, is she is taking soils from each of these sites and planting into them a number of different hairy vetch types or cultivars, accessions that have been collected from around the world. And this is a project that we're carrying out in conjunction with Dr. Jude Mall, who I know has done a webinar in the past through eOrganic. Where he has looked at these different populations of vetch and have determined that there are great differences in their genetic makeup, of the plant's genetic makeup. And we want to see if by having different types of vetch that are planted into these soils, if that's going to impact the Rhizobia that associate and form nodules with each of them. So NAPI, in her research, took these different varieties of hairy vetch, sterilized the seeds, and then planted them into a sterile mixture of sand and vermiculite, and then inoculated them with soils from each of these locations. So thereby we're ensuring that the only nitrogen that these plants are going to get is fixed through the bacteria that we've added in the soil, and that if the plants grow well, we know that they're fixing nitrogen. If they die, well, we've determined that they probably didn't have the right Rhizobia present to do their thing. And here's an example of that case. We're finding that in most of the farms, actually all of the farms, that when there is a history of hairy vetch in the field, if that soil has enough Rhizobia in it to carry out effective nodulation and nitrogen fixation with the varieties under study. And in many cases, we're finding that there's just not enough Rhizobia present in fields that have not had hairy vetch for the plants to grow well. So it seems that in our preliminary results that having a history of hairy vetch cultivation does make a difference in who is actually present in that soil. We can also look at number of nodules to determine this, where we have the 3 sites, and in each of the sites with a hairy vetch history, there are significantly greater numbers of nodules present than in fields that were, soils was taken and used to inoculate plants where there was no hairy vetch planted in the past. Now just for a little bit, this is the last gel I'll show, I promise, but this is a way that we look at the actual bacteria themselves to determine how the bacteria present in these positive vetch and negative vetch fields might be different from one another. And we saw right away that there are differences in what we call the genetic fingerprint of these Rhizobia from fields that have seen hairy vetch many years in a row from fields that do not, where you notice that in each of these is a different type or strain of Rhizobia pulled from those fields. It seems like there's just one type of Rhizobia present in these fields that didn't have hairy vetch, where fields that did have hairy vetch have a wide variety of different types of Rhizobia that are present. So with that, I'm going to end this and open it up to questions, but I hope this in general has given you some tools you can use on your own to look at nitrogen that's fixed in your crops, and look at how nitrogen delivery from cover crops can be used to augment your the amount of nitrogen that's being added to your system instead of using otherwise purchased cover crops, nitrogen sources. So we do have a website where if you're interested, you can keep track of what we're up to. And at this point, I will open it up to any questions. Thank you.
Thank you very much, Julie. We're about to start our question and answer period, and for those of you who missed the beginning of the presentation, you can use the question box on your screen to type in questions and hit return. If the question box is closed, you can click on the small plus sign next to the word question to open it up. We'll be reading the questions out loud, and we'll answer as many as we have time for. For those of you who might have to leave earlier, this webinar will be posted on the eOrganic website within the next 10 days or so, in case you'd like to hear it again or share it with others. We'll also be posting the slides as a PDF on the archived webinar page on eOrganic. And I'll put the eOrganic website address up on the screen momentarily. Here it is, at www.extension.org/organic_production. If after the webinar you have additional questions or questions that haven't been answered, you can use the online Ask an Expert system at extension.org/ask and you'll get an answer. And finally, we value your feedback, so we'd very much appreciate it if you could fill out our follow-up survey, which you'll be receiving in an email. So now let's move on to the questions. We're getting quite a few questions coming in. First question, which part of the plant, the roots or shoots, contributes the most nitrogen when decomposing?
That is a wonderful question. You can think of nitrogen in a plant as being the stuff that makes it green. So nitrogen is used by the plant to make chlorophyll, among many other things, but chlorophyll is made up of a good amount of that nitrogen. So if you notice in that picture I had of the corn where it was showing we had the nitrogen that wasn't as available as under the hairy vetch mulch, and you saw how it was yellow. So that's if your crop is yellow, it's not getting enough nitrogen. So most of the nitrogen is in the shoots where they're green, and the roots only can contribute, or on the total amount of nitrogen in the plant, about 10 to 20% of the total nitrogen in those roots. We're actually just starting a project now in our lab to try to look at, to answer your question directly, to try to look at how much nitrogen is contained in the root system, because of course when we measure this and we take off just the shoots, we're only taking the top portion. So we're completely missing that below ground nitrogen, although it's less, that below ground nitrogen that is contained in that cover crop. So yes, it is there, and right now it hasn't been measured in many of these species, so we're hopefully going to have an answer to that question more specifically in the next year or two.
Okay, the question, can undersown clover in corn provide nitrogen, or would the clover have to have decomposed first?
That's another good question. I get asked that one a lot, not necessarily with clover, but just in thinking about intercrops of corn or a non-legume crop with a legume cover crop. And the answer is, where I said that nitrogen doesn't leak out through the nodules, there's a little tiny bit that will come from the nodules, but a majority of it is in the shoots. So if you really want to get that nitrogen from your undersown clover, you should find some way to mow it or get rid of that shoot material so that it can decompose in the soil. Along with the little bit that is released during active fixation, which again is a very, very tiny proportion of the total nitrogen fixed. Along with that amount, you will have nitrogen that's released through nodule death over time. I mentioned that the maximum nitrogen fixation point in a cover crop is right before or at flowering. So that's when it reaches that usually between upper 3%, 4% of the crop or the cover crop biomass being nitrogen. And if you kill it at that point, then you'll have the most nitrogen released into your system. So at that point, because the legume cover is putting most of its energy into forming the seeds, it stops feeding the nodules. It stops giving the carbohydrates to the nodules, and the nodules will die. And through that death of the nodules, you will have a little bit more nitrogen that's released at that point. But the majority of it is in the biomass, so I would find a way to somehow get rid of that biomass and put it back into your soil.
Okay, next question. What were your seeding rates for monocultures and the rye mixes?
That is a really good question, and I would need to look that up. Is that a question that maybe could be, I believe it was between For the monocultures, it was 23 and 28 pounds per acre. But to get an actual value, I'd like that question to be asked through the Ask an Expert, if that's okay.
Okay, maybe we can post that information on the registration page
good.
afterwards.
So I can look it up. I just don't have it in front of me right now.
Okay. What about optimal kill of cover crops with rolling date along with most nitrogen produced?
Can you repeat the question, please?
Well, I'm not sure. What about optimal kill of cover crops with rolling date along with the most nitrogen produced? Do you understand the question?
I mean, I think I do. If the question is asking when we killed the cover crop, so the optimal kill time was the point at which the optimal amount of nitrogen is being produced. And if that's the question, then yes, we found that, especially with hairy vetch, the time at which we could kill the cover crop with the roller was in late April, early May at the earliest. And that, when looking at the amount of nitrogen contained in the biomass at the 3 roll dates, that was the point at which we did have the highest amount of nitrogen. So after that point, when the cover crop was a little bit more mature, there was less nitrogen contained in the biomass, likely because it had shuttled most of it away from the leaves and into the flowers and the seeds.
Okay. How accurate are the nitrogen fixation rates when they're plowed under rather than rolled?
How accurate are the rates? So
I'm not sure I understand the question. You feel free to type it in.
Yeah, well, I think if we're, so the rate, I don't know if it's referring to the actual fixation rates, which would be the amount of nitrogen fixed, but the plowing under or the rolling would be less related to the amount of nitrogen fixed as it would be to the release of that nitrogen into the environment. So if you have a cover crop that has a certain amount of nitrogen in its biomass, when it is incorporated, you're going to have a much faster release of that nitrogen into the soil. Usually it is a rule of thumb that if it's incorporated, you get about half of the total nitrogen in that crop being released over the growing season, where if it's just mowed and left to sit on top, you get about 40% of that nitrogen. And that makes sense, because if you think about it, you have a cover crop that when it's incorporated or mixed into the soil, you have the more of a soil to residue contact. The microbes can go about their thing and break down the cover crop biomass, releasing that nitrogen into the soil from all the biomass, because all of it is touching the soil. Where if you just have the mulch sitting on the soil surface, only the bottom portion of that mulch is going to be in contact with the soil that's doing the microbes that are doing that decomposition. So it's going to be a little bit slower. And we actually have some experiments in currently, so we just planted them a couple of months ago, where we are going to look at different ways to terminate these legumes. And we're looking at 4 legumes. We're looking at Austrian winter pea, the balansa clover, which is one of the unique ones that some growers in North Carolina are really interested in. So Austrian winter pea, balansa, hairy vetch, and crimson clover. And we're going to terminate them this spring in different ways. So we're going to roll them. We're going to mow them and just let the residue sit on the soil surface. And we're going to mow them and mix it into the soil. And then finally, we're going to burn them down with herbicide. And in all these treatments, we're going to compare the rates at which nitrogen is released to a subsequent corn crop, with that assumption that any residue that's in more direct contact in the soil, such as the treatments that are incorporated, will release the nitrogen faster. And we'll get different patterns that emerge of nitrogen release from those different legume termination treatments.
Okay, here's a question about crimson clover. Crimson clover is often touted as the best nitrogen fixer early in the spring. Your study focused on nitrogen fixation late in the spring and found that crimson clover immobilized nitrogen. Do you have any data on crimson clover versus hairy vetch early in the season?
There's some, I don't have it in front of me now, but there are papers by Michael Wagger, who is in this department here at NC State, Soil Science Department. coming from the 90s that have rates of nitrogen fixed by crimson clover earlier in the season. So that does exist, but we have not done it recently, and we don't know anything about release rates when you kill it earlier in the season using something such as the roller. So it's out there, definitely.
Okay. How does a clay soil versus sandy soil play into the nitrogen release? Faster or slower?
We don't have data on that. That's a good question.
Okay.
That's something I'm unable to answer at this point.
Is there a minimum viable spores per seed that should be used when inoculating legumes?
Okay, so inoculation, again, is that process through which you're adding the right bacteria to the seed. And when you buy it, so if you go and you buy legume cover crop seeds, you buy the seed, and then they say, oh, do you have an inoculant? And they sell you an inoculant. It's usually a bag of inoculant, which is just a peat material that contains living bacteria, living Rhizobia. Usually costs a few bucks, so it's not expensive. The minimal amount, I can't give an actual value of the number that of viable Rhizobia that are necessary, but it's not much. You can have strong nodulation with only a few viable cells per seed. But in thinking about the rate, a lot of what you're putting on is peat. And so especially if we don't know the number of viable or living cells in that peat, if it's older, or you've had it for a couple of years, or it hasn't been in a fridge, things like that will all affect the number of viable Rhizobia you have that will be able to form those nodules to start that process of nitrogen fixation on your seed. So it's really going to vary.
Okay. In the soils with no hairy vetch history, were the hairy vetch plants nodulated?
Yes, they were, but some of them, well, not actually, it's not true. Not in all of them. We had some that were nodulated a little bit, but most of them did not grow very well. So there was definitely an effect of not having the numbers necessary to form nodules on those plants when there was no history of hairy vetch.
Okay. How could this be relevant for conventional farmers seeking to reduce their use of synthetic nitrogen?
Oh, well, this is extremely useful, I would say. We've been talking here with some economists and trying to figure out how we can make this less expensive of a management tool. Because right now, here in North Carolina, if I remember correctly, a majority of the price that farmers were paying in carrying out the whole process of planting a legume cover crop. So that would include getting the seed, the inoculant, the fuel for the planting, etc. The majority of that was eaten up by the legume seed itself, because you get a lot of our legumes, especially the hairy vetch, we get it from the West Coast. So we've been toying with the idea and trying to encourage farmers in this area to produce a less expensive local seed so that farmers, conventional or organic, can have a source of legume cover crop seed that they could use and thereby bring their prices down by quite a bit. Right now, I haven't looked at the price of nitrogen recently, but last year I remember that it was still a lot cheaper for conventional growers to buy synthetic nitrogen than go through the process and the cost of planting a legume cover crop. So I think when nitrogen prices increase, which I predict will happen in the future as we approach and face issues with energy shortages, etc., that we're going to have nitrogen prices increase greatly and maybe see more conventional growers start to consider legume cover crops as a management tool to contribute nitrogen to their system. It's not a new thing to study. If you look back in the literature, of course, from the 40s and 30s, 20s, before nitrogen fixation, nitrogen fertilizers really took off as an input, nitrogen fixation was studied quite a bit, and it still is studied quite a bit in developing countries, where it can be the primary tool that is used to deliver nitrogen to systems where farmers just don't have the money to buy nitrogen fertilizers. Right now, one of my graduate students, actually the one who I had presented the roller crimper data from, she is getting ready to start a project in Malawi looking at how we can get more bang for our buck in using legume cover crops that are edible and trying to increase the amount of nitrogen that's being fixed by these legume cover crops so people in poor regions can get something that they can eat, a protein-rich legume seed, but also, or bean, but also have that crop or that plant produce enough nitrogen that it can go back into the soil and help alleviate some of the really serious soil fertility issues that Africa is facing right now.
Okay, have you looked at flail mowing versus rolling to see if nitrogen residue is more readily available?
Okay, well, I think I just answered that. That's going to hopefully be answered by our upcoming work.
Okay. Is it a no-no to incorporate cover crops into soil instead of crimping? I realize protection against weeds would be lost.
Yeah, no, it's not a no. It's not a no-no. It's a there have been a lot of efforts recently in trying to create what we would call no-till organic systems. So trying to design management systems for organic crops where we don't need to till in the soil or till in the cover crop or till up the soil, because every time you till the soil, you're losing a little bit of organic matter. You turn over the soil and the microbes go crazy because they get all this new oxygen and they start to decompose your already existing organic matter in the soil. So you don't want that. You have a great decrease in soil quality by multiple tillage events in a year. So there's been a great effort in this area to try to reduce tillage, but I wouldn't necessarily call it a no-no because it in part depends on when you want that nitrogen to be made available. And again, hopefully we're going to have some data coming up in the future looking at when that nitrogen is released relative to rolling, and if we can start to match the timing of when that corn, in our case, needs the nitrogen compared to when the cover crop is releasing it upon its germination.
Okay. If vetch or other legumes have higher measured nitrogen earlier in the season, where did the nitrogen go? Could it just be that you did not collect leaves that had dropped off the plant or already started to decay?
Yeah, we had that same question, and we went back and looked at the data, and when we were collecting, especially with crimson clover, we showed that happen. So what the asker of the question is referring to is we saw a decrease in nitrogen and biomass, in particular, in that second roll date. So where did the biomass go? And we did our very best to collect everything, and in looking at the nitrogen, we think that it was so far matured, in the case of the crimson clover, that a lot of that nitrogen had been taken out of the leaves, channeled into the seeds, and it could be that we didn't get all the seeds, or it wasn't distributed well enough in the biomass to be able to measure. But we did have that same question, and we're still not 100% sure what happened, but we're pretty sure we collected as much of the leaf material as we could upon that measurement.
Okay. The hairy vetch rye mix had a higher carbon-nitrogen ratio, which should tie up nitrogen, but the corn crop looked better. How is that?
Can you repeat that question?
Okay, yeah. The hairy vetch rye mix had a higher carbon-nitrogen ratio, which should tie up nitrogen, but the corn crop looked better. How is that?
Yeah, that is a question that we are pondering ourselves, and you're referring to that last picture I showed of Jonathan standing next to that beautiful, tall, green, lush corn. And it's pretty well known that when you have a mix of a legume and a non-legume or a grass, that the grass sometimes can take up nitrogen from the soil, so reducing the amount of available nitrogen in the soil, because now the grass has it in its own biomass. And then the legume says, oh, well, I don't have a lot of free soil nitrogen, so I'm going to fix more. So we're still exploring these ideas a little bit, but we really would like to know how much nitrogen was being fixed by the legume when it was in mixture with the rye. So in the data I gave, we looked at a combined sample of the rye and the legume, the vetch, mixed together. So the sample was diluted, I guess you could think of as that. In that way, diluted by the non-legume biomass, the rye biomass. But I would really like to know if there was more fixation going on, so there might be more nitrogen in that legume that could have been helping to drive some of those effects. But I think it might not just be due to the legume as well. There might have been other effects that helped that corn grow well, such as improved moisture retention if there was an incredibly thick mulch on that surface. Other things that would help that corn get as big, tall, and green as it was. But we were very surprised by that as well.
Okay. Do you know if there was any nitrogen loss, leaching, or denitrification from the cover crop decomposition?
We don't, and that's another thing that we're going to be measuring in this upcoming study where we're looking at the different termination treatments. We would really like to get a sense of the entire nitrogen budget and where things are going, how things are moving. So the only pool really that we've been assessing is this flux of nitrogen from the legume cover crop into the surrounding soil. But the person who asked the question is exactly correct, that we could have other losses of nitrogen that are taking place during the decomposition of that cover crop, and we would really like to get a handle on that. But it's very hard to measure all those things in one go around, one season. So it's on our list, our wish list of things to do, but we haven't quite gotten there yet.
Okay, we have time for one more question. For smaller scale without the equipment that you have, like a roller crimper, what are recommended options to use? Example being a smaller scale farm or home garden.
Yeah, for home gardeners, what's often done is the removal, because the mass can be so thick of cover crops once they're killed, it's hard to plant into that on that material. So what's often recommended is that you take the cover crop material away, compost it, and then bring it back. So you're sort of consolidating the nitrogen into a more manageable material that you can then put back onto your plot. But there are other examples that I think you can find pictures online for some of these through the Rodale Institute Roller Crimper Gallery. There's an image where there's a young woman who has a board with metal stakes or spikes attached to the bottom of it, and she steps on the board and crimps, so to speak, crimps her cover crop in that way. So if you really want to do the crimping, that's one way that it can be done. And I haven't done that myself, so I'm not sure how that would work, but I know it's done elsewhere.
Okay, well, we're running out of time, but I'd like to thank you all for your questions. And as I mentioned before, if you have additional questions, you can use the eExtension Ask an Expert system at extension.org/ask. This webinar will also be posted on the eOrganic website within the next 10 days at extension.org/organic_production, and please fill out our follow-up email survey. Thank you so much for coming again today, Julie, and thank you all for joining us.
Thank you.